Alright, hi everybody. Just want to give you a couple unusual cases of what can happen during meiosis with some chromosomes. So if we break this down, chromosome abnormalities basically deal with two things. Either having an incorrect number of chromosomes, which we call non-disjunction, and basically what happens here is the chromosomes just don't separate in the patterns that we learned about in meiosis. Or we can have breakage of chromosomes, and we'll see how chromosomes can vary a little bit based on where they break and what happens to that information. So let's start with non-disjunction first. So basically what we got here is we've got issues with spindle fibers that causes separation problems. So you can have separation issues that happen in either meiosis 1 or meiosis 2. In meiosis 1, what would happen here is your homologous chromosomes do not separate properly. And in meiosis 2, what could happen is your sister chromatids don't separate properly. And both of these are caused by spindle fiber issues. And your result, what you end up with, is either too many or too few chromosomes. So if we look at this picture, it shows meiosis 1, meiosis 2, and then the gametes that are produced. And you'll notice that there are some problems, and you can have non-disjunction occur in a couple different places. If you look down at the bottom, these products down here, with some that are too few or too many, they all have an incorrect number in those gametes. And the reason for that is an error in meiosis 1. So what we're going to see when we compare with what happens in meiosis 2, here you only have half the amount with the incorrect number. The other half still is normal. So an error in meiosis 1 is a little more detrimental to the gametes' chromosome numbers than an error in meiosis 2. And we've got some names depending upon how many chromosomes are present. Trisomy, and you'll see the root word tri in there, means that cells have 3 copies of a chromosome. Where monosomy, and you see mono in there meaning 1, shows that the cells only have 1 copy of a chromosome. And I've got some examples showing those little colored chromosomes down at the bottom. Now, chromosome disorders in humans actually has a pretty high frequency, and the reason why we don't see people walking around with many of these chromosome problems is because most of the time it's so disastrous. The alterations that happen are so disrupting to the sequence of DNA is that the embryos don't even go beyond maybe a couple cell divisions. Those that do go beyond a couple cell divisions and develop into a fetus typically exhibit some developmental problems, and that's really due to some biochemical imbalances. Could be a hormone issue, could be some transcription factors, depends on exactly what chromosomes we're looking at, and based on this, some conditions can be tolerated. If you upset the balance the least, that's kind of survivable. But there are some sets of characteristics that we see, and that's why we call it a syndrome, because you can kind of go through a checklist of phenotypic things that will happen. So one of the most popular and one of the most easiest to talk about is Down syndrome. And by easy, I mean it's just an extra copy of the 21st chromosome, and that's why it's called trisomy 21. Statistic-wise, about 1 in 700 children born in the U.S. have this syndrome. And although chromosome 21 is the smallest chromosome, it can still have very severe effects. And we actually have data that supports that the age of the mother has a correlation to Down syndrome. And as you look at the graph, you can see that as you get older as a mother, you have a higher risk of having a child with Down syndrome. One of the things that we think is that there's mutations that build up over time in the mother's egg cells where those spindle fibers do not act normally as they should. One of the ways of testing for some of these syndromes that I'm telling you about is something called an amniocentesis. It was very risky, and today still is probably considered very risky, only used in high-risk cases. And you can see the statistics in the 70s, about 1 in 200 pregnancies women who went through an amniocentesis would miscarriage due to that, where 2006 data is about 1 in 1,600. So we have improved the procedure, and this is what the procedure looks like. They do use a syringe with a very long needle, and they will go through the womb into the amniotic fluid and take some fetal cells, because that fetus inside the mother is shedding skin cells just like we're shedding skin cells sitting at our desks. This is typically done in the 2nd trimester. And they'll take a karyotype of those cells, and that's where they'll look for extra copies. Now, what the karyotype initially looks like is pretty messy. It's just a great big smear of things. And using some computer programming, we can go through and line all those chromosomes up. And in this case, this one was a boy. Now, what gets real interesting is when we start looking at sex chromosome abnormalities. Alright, so these differences that we're going to look at in the sex chromosomes, these happen basically in how you inherit your X's and your Y's. And so there's a variety of very distinct syndromes that we have. We're going to talk about 4 of them, which are listed there in red. So the first one is something that's called Klinefelter syndrome, and it's the case of having an extra X. Phenotypically, this looks like a male. However, they are sterile, and there's some issues with the hormones communicating with having extra X and Y chromosome hormones. So what you find here, typically there are some feminine characteristics, very tall height, but normal intelligence. And I'll touch on the intelligence piece here in a second when we start looking at a couple more different syndromes. Accompanying all of these, I do have karyotypes, so you can see what those look like. Having an extra Y chromosome, again, phenotypically looks like a male, and this is something we call Jacob's syndrome. We do see a slightly taller height with this also. Normal intelligence with some learning disabilities, delayed emotional maturity. One of the things that we originally thought was that males who commit very violent crimes, we thought that possibly they might have this as a genotype, maybe giving them an extra boost in testosterone. And there was lots of studies done on inmates, probably a little on the unethical side in looking at this, and as of today we have yet to find a connection to having an extra Y chromosome in a violent crime. The next one I want to talk to you about is Trisomy X. This is having 3 X chromosomes, produces healthy females. Nothing phenotypically wrong here. And the reason why is something called a bar body. Early on in the development of a zygote, one of the X chromosomes becomes inactive. And in this case, 2 of them will become inactive. So you kind of get a range of different X's that are expressed. I know you might have a question, what if one of the X's carries a trait for a sex-linked disease? As long as you have a normal X expressed in some of the cells, it would mask that other allele. But this is the case for calico cats in that they are always female, and how you get that different pattern of coloring in their coat color is based on which X is activated or which one is inactive. Last one to look at is Turner syndrome. In this case, the individual only inherits 1 X chromosome. It's got a very degree of effects, but one of the things is that you never fully mature in some of the characteristics that we look at in going through maturity and going from adolescence to adult. These are also typically sterile individuals because they don't have the correct amounts of hormones to allow all of the development that needs to happen in order to make you fertile. So those are kind of all of our characteristics with whole chromosomes that I wanted to talk to you about. We can also change the structure in the chromosome. So these are pretty easy to look at. Their terms really do mean exactly what happens, but I will tell you that there's two events where these errors happen at. One's in replication and one is in the process called crossing over. So in replication, you could delete sections, like if, for example, if you look at that top one, letter D is deleted, or you could duplicate it, and you'll see BC is duplicated, where you just repeat a segment. Now, obviously, we know that on these segments we have coils of DNA wrapped around that, so you are impacting the genetic code. In crossing over, we're going to look at how we can switch segments of DNA. We can have an inversion where it reverses the segment, or you could have translocation where one segment moves from one chromosome to another. And this would be a chromosome that's not related to the homologous chromosomes that are lining up. So for example, it would be information from chromosome 1 moving to chromosome 2. Now, all the things that we've talked about, having extra chromosomes or extra amounts of genetic information on your chromosomes, has been very specific to animals. In plants, however, it's very common to have extra sets of chromosomes. And what will actually happen is the more chromosomes that you have, the bigger the fruit is produced. And strawberries are a good example of this. Most strawberries that you eat today from the grocery store actually have 8 copies of their chromosomes compared to the wild strawberries that have just maybe 2 or 3 copies. So it's something that has been genetically modified, has been bred into those plants. And just something to end on in looking at the different types of systems that determine sex. You have an example of the XY system. That's what we've talked about today. Insects follow something a little different called the XO system. To be female, you have 2 X's, but a male, you only have an X chromosome. Chickens are a little bit opposite of us. Having just 1 Z chromosome gives you a female chicken, and 2 Z's give you a rooster. And then insects are very interesting, specifically looking at the honeybee. Having 2 copies of the chromosomes, that's what makes you a queen bee. When you become a worker bee, however, you only have half the amount of chromosomes. You're a haploid organism. And in this example, the bee is completely normal, developing with half the amount of genetic information. All right, as always, bring any questions that you have to class.